Welcome to the deep dive. We're jumping straight in today, looking at one of the really big hurdles in early CRISPR work. How do you stop that Cas9 enzyme, the cutting part, from snipping DNA in all the wrong places? And the fix? Well, it was kind of counterintuitive. It wasn't about making the guide RNA smarter. It was actually about making the Cas9 enzyme itself weaker.
Exactly. Intrinsically weaker.
Well,
The researchers figured out that if they could sort of fine-tune the enzyme's grip, specifically making its non-specific connection to the DNA backbone a bit looser, they could engineer this new version, pCas9-HF1, HF for high fidelity. And it basically wiped out detectable off-target cuts genome-wide, while crucially keeping the on-target cutting working well for most guides.
okay, so let's just set the scene a bit more. The challenge was the standard Cas9, the one from Streptococcus pyogenes, it was just, well, sloppy. It would cut sequences that just looked similar to the target, not only the exact match, which is obviously a massive problem if you're thinking about high-precision research or, therapies.
Absolutely disastrous for therapies.
Yeah.
And people have tried fixes before, right? Like shorter guide RNAs or using two Cas9s to make single-strand breaks, paired nicases. But these were often complicated, fiddly, and you usually lost efficiency, or it just wasn't easy to implement. What was really needed was a simple, drop-in replacement.
Right.
And the key idea was this excess energy hypothesis. Basically, the Cas9 and its guide RNA stuck to the DNA a bit too strongly overall. That extra energy meant it could tolerate mismatches and still cut. So the goal became, how do we dial back that nonspecific energy?
Okay, so how did they actually do that? Dial it back.
Well, they looked at the protein structure, the 3D shape, and identified 4 specific amino acids, N497, R661, Q695, and Q926. These spots seem to be making hydrogen bonds with the DNA's phosphate backbone. That's a nonspecific interaction, just holding on. The hypothesis was if they swapped these out maybe with alanine, which is pretty non-reactive, they could reduce that general stickiness without messing up the part that recognizes the actual target sequence.
Okay, rational design. So they made the changes. They created this quadruple mutant CPAS9-HF1. Big question then, did making it less sticky just break it? Did it still cut where it was supposed to?
That was the crucial test. They used an assay in human cells looking at disruption of a green fluorescent protein gene, EGFP. And the results were pretty compelling. HF1, the four-way mutant, it kept strong on-target activity. Across 37 different guide RNAs they tested, for 86% of them, that's 32 out of 37 HF1, worked at over 70% the level of the original wild-type Cas9. So yeah, they managed to make it choosier without crippling its main function most of the time.
Okay, hurdle 1 cleared, it still works. Now here's a point to think about for you listening. If they just reduce the overall binding energy, you might expect fewer off-targets, right? A significant drop. But did the researchers just see a decrease, or was it something more dramatic than that?
It was definitely more dramatic. They used a technique called Guide-E-Sec. It's designed to find all the cut sites across the entire genome, unbiased. They tested it with 8 different guide RNAs targeting various human genes, wild-type Cas9. For 7 of those guides, it made between 2 and, 25 off-target cuts.
Okay, so quite a few.
Yeah, but mucicas9-HF1, for 6 of those 7 guides, 0 detectable off-target cuts, none. Across the whole panel for those guides, only 1 single off-target site popped up that wasn't there for wild-type 2. It was basically clean.
Whoa, okay, so not just fewer, but essentially gone.
Yeah.
Below detection limits.
Exactly. Exceptional precision. That subtle energy reduction was enough to push nearly all those incorrect sites below the cutting threshold.
But what about the really tricky targets? Like repetitive sequences, those are usually hotspots for off-target mutations, right? A real stress test.
Right, the ultimate stress test. They picked a guide RNA, VGFASA2, that was known to be super promiscuous. Wild-type Cas9 made 144 off-target cuts with this guide, SPCAS9-H1. It eliminated 123 of those. Still some left, but a massive reduction. And for another tough one, VGFA site 3, wild-type had 32 off-targets. HF1 got rid of 31 of them.
Okay, so even under maximum stress, it's a huge improvement.
A huge improvement. It showed the principle holds up even when the system is really pushed.
So stepping back, the real innovation here seems to be the simplicity, right? SPCAS9-HF1 is basically a single protein change. It's a plug-in replacement. You use the same guide RNAs, you just swap the Cas9 enzyme. No need for complex setups like paired nicases or those other fusion proteins.
Precisely. It avoids all that complexity, the design constraints, the potential efficiency hits of the older methods, and it validated this whole strategy. Tuning non-specific protein DNA contacts. That's a general recipe you could potentially apply to other DNA-binding proteins, other CRISPR systems maybe. This really set a new benchmark for specificity. It made precise editing much more practical for basic research, functional genomics, and crucially, it started to de-risk things for any potential preclinical or therapeutic work where off-target effects are just, well, unacceptable.
Okay, let's quickly summarize the key takeaways from this deep dive on CEPI-Cas9-HF1. First, the high-fidelity version came from rationally changing just 4 amino acids known to grip the DNA backbone non-specifically.
Second, reducing that nonspecific rip worked incredibly well. It pretty much eliminated detectable off-target cuts while keeping good on-target activity for most sites tested, around 85% retention.
And third, this gave the field a simple single enzyme solution for high specificity, a huge step up from the more complex fixes that came before.
Yeah, it was a big deal. Now, the paper's claim is no detectable off-target effects, which sounds amazing. But you always have to remember, detectable depends on the sensitivity of the methods used, right? This was Guy-DSEC back in 2016. So sort of provocative thought is, could there still be ultra-low frequency mutations happening below the detection threshold, even of today's best methods? That quest for absolute 100% certainty, it's probably never truly over.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has 3 main goals. 1, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2, to build a supportive community where we can exchange the most valuable experience-based research advice. 3, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.